So this morning I want to talk about some project that I've been involved with for some time related to the CNS pathology of myotonic dystrophy. First of all, I have no financial relationships to disclose, and I'm certainly not going to talk about any drugs. So what sort of the goals or objectives of this are to have you be able to identify the molecular mechanisms of myotonic dystrophies type 1 and 2, have some familiarity with the neuroimaging findings that I'm going to talk about, and then evaluate what's known about the correlation between the clinical features of the disease imaging and the pathological studies of the CNS that have been done in the myotonic dystrophies. So we'll start off just with an introduction to the disease process, and then talk a bit about molecular pathogenesis, and then what's known about the neuropathology, and then a little bit about what we're doing and trying to do in the future. So myotonic dystrophy is actually one of the most common of the inherited muscular dystrophies. It's the most common with an adult onset, and it has a prevalence of somewhere around 1 in 7 or 8,000. It's an autosomal dominantly inherited disease, so it tends to run fairly strongly in families. And there is a tendency often for anticipation, like pattern, where the children frequently have an onset that's earlier in life than the affected parent. So when we talk about myotonic dystrophy, we usually think of it as a muscle disease. I mean, that's what it's named. It's called myotonic dystrophy. And myotonia refers to the inability of the muscle to relax. So like when somebody shakes with myotonic dystrophy, if they shake your hand, you have a friend for life because they can't let go. So the contraction is difficult to stop. But coupled with that is weakness and muscle atrophy over time. And it has a tendency to affect certain muscle groups, particularly muscles of the neck and face. There's often wasting of the sternocleidomastoid muscles in the neck. Temporalis muscle often is wasted, and often you can see almost sort of a concavity on the lateral aspects of the upper face. Unfortunately, it also can affect respiratory muscles, and this is often what leads to the death of patients with more severe disease, is that they can have respiratory weakness, both of the diaphragm and of the accessory respiratory muscles, that can then frequently lead to aspiration pneumonia. In addition to the skeletal muscle problems, they have other problems, and one of the most frequent things is to have cataracts. And the cataracts often come on quite early in life and have a very characteristic appearance that I'll show you in a few minutes. In addition to skeletal muscle problems, they also can have cardiac problems, including predominantly conduction effects with cardiac arrhythmias. Frequently have premature frontal balding, which can sometimes give them a facial appearance that you can recognize quite readily. In addition, they also have insulin resistance, so type 2 diabetes is very common in these patients, and the males can have testicular atrophy, particularly the more severely affected. Smooth muscle, it doesn't escape the disease. It does affect the GI tract, and there can be problems with gut motility and sphincter laxity. One of the things that's been sort of underappreciated, I think, by many people, but people who take care of these patients recognize that many patients with myotonic dystrophy, as they get older, develop problems with cognition. They also have problems frequently with hypersomnia. The cognitive impairment is somewhat different than typical AD, typical Alzheimer's disease. They don't necessarily have a lot of memory problems, but they tend to have more problems with executive function. They can have visual spatial problems and sort of a kind of a abulic, apathetic type affect. There is a congenital form of myotonic dystrophy which is associated actually with frank mental retardation, but most people with myotonic dystrophy do not have cognitive impairment, particularly early in life, but it seems to be more of a neurodegenerative-like process with the cognitive impairment coming on later. There are two basic types of myotonic dystrophy. By far the most common is what we call DM1. We tend to call it DM because it's dystrophia myotonica is the old name for it. So when I refer to DM1, DM2, that's where that's derived from. About 98% of the patients have classical DM1. Most of them have adult onset, but there are some that have a congenital form. DM2 is considerably less common. It has a slightly different clinical appearance, but there's many shared features, and it typically is somewhat milder, and they do not have a congenital form. And we'll get into the genetics of these in a bit. So this is a family of patients with myotonic dystrophy. There is a grandmother, a mother, and a child. And the grandmother had a later onset, the mother a slightly earlier onset, and the child had essentially congenital onset. And this is a pattern that is not infrequently seen, so that there is a so-called anticipation, so that as the gene is passed from one generation to the next, there seems to be an earlier onset and a more severe form of the disease frequently. Unlike in the polyglutamine diseases like Huntington's disease and many of the cerebellar ataxias, this is actually more commonly associated with maternal transmission, where you see the jump rather than paternal transmission. This is an example of the cataracts that are seen. They're called Christmas tree cataracts. This is a slit lamp exam, and you see these sort of multicolored refractile pattern in the lens. And these often develop in the third decade of life, so these come on quite early in life. These are not sort of senile cataracts, but they come on. It may be the first manifestation of the disease, and in some patients with mild forms of disease, it may be the only manifestation. The muscle pathology is somewhat characteristic, although not entirely unique. There tends to be a lot of central nuclei in the myofibers, some myofiber variability in size. Can't seem to find. There are so-called ring fibers, and there's often these nuclear clusters as well, but not a tremendous amount of endomysial fibrosis that you might see in some of the more severe forms of muscular dystrophy. The DM2 is a little bit different. They tend to be milder. They have a slower progression. The weakness usually starts more proximally than it does in myotonic in DM1, and there's often sparing, particularly of the lower extremities, so the calf muscles can become very hypertrophic, somewhat similar to what you see in Duchenne's muscular dystrophy. They also can develop cataracts. They do have involvement of the endocrine system of the CNS and gonads, but it's usually somewhat milder than it is in DM1. There is no congenital form of DM2, and there seems to be less anticipation. So let's talk a little bit about the molecular pathogenesis. So the gene for DM1 was originally isolated back in 1992, and it was shown to be trinucleotide repeat expansion. It's a CTG-repeat. It's in the terminal exon of the DMPK, which is the dystrophy myotonic protein kinase, or myotonin. So this is in the 3' untranslated region. So this does not make protein, or at least we didn't assume that it made protein, at least initially. And there is some somatic instability of the repeat, so the repeat size can be quite variable throughout the body. And of course, there is some generational instability, which probably accounts for some of the anticipation that is seen. The gene is located on chromosome 19. There's a couple of flanking genes, the DMWD and the 6'5" gene. And there was some interest in whether these genes may have played a role in the pathogenesis. We'll get back to that in a second. So the clinical picture seems to correlate reasonably well with the repeat expansion size, as is the case with a number of other these trinucleotide repeat disorders. So if you have fewer than 50 repeats, you're essentially normal. And most people have fewer than 35 repeats. In the range of 50 to 80, you can have mild symptomatology, mostly cataracts. But there's a greater likelihood that subsequent generations will have a greater number of repeats. So your children may develop more full-blown myotonic dystrophy. Between 80 and 1,000, there's basically the full range of mutation, usually adult onset. And if you have over 600 repeats, you tend to have adult onset, but you have a greater likelihood of your child having congenital onset. And typically, if there are over 1,000 repeats, then it's most likely to be congenital onset, although there are a few cases of adult onset that are greater than 1,000. The congenital form is really much more severe. They tend to have mental retardation. The brain doesn't develop completely normally. It hasn't been terribly well studied pathologically. They do have fairly widespread cerebral atrophy and often hydrocephalus. They have other problems, including orthopedic problems. And as I said, the majority of cases have repeat lengths greater than 1,000. So what are the possible pathogenetic mechanisms of this mutation in the myotonin gene? So one would just be that it's a loss of function of myotonin. And there is some evidence to suggest that perhaps in patients with myotonic dystrophy there is some reduction in the activity of this enzyme. But if you knock this gene out in mice, you don't really get a phenotype that in any way resembles myotonic dystrophy. Likewise, these flanking genes that could possibly be affected by this repeat, if you knock those genes out, you also do not see a phenotype. So it's unlikely that either of those two mechanisms play a major role in the pathogenesis of the human disease. So one of the observations that was made fairly early on is that these patients who have this mutation develop repeats of double-stranded RNA that contain the CUG transcript. And this can form a hairpin configuration. And these tend to aggregate in the nuclei of muscle cells and actually has been shown later in cells in the brain as well. Here's an example of these RNA inclusions within, these are in myocytes in the nuclei. And then about 12 or 13 years ago now, John Day and Laura Ranum here at the university were studying some patients with a more atypical form of myotonic dystrophy, the DM2 patients, and they found that even these patients have a completely different mutation. So there is a mutation in the zinc finger protein 9, which is located on chromosome 3. And this is also a repeat expansion, but it's a tetranucleotide repeat. It's a CCTG repeat, and it's in an untranslated region, actually within an intronic site. But yet these patients have a very similar phenotype to patients who have a mutation in a completely different gene in a completely different site. So the normal gene for the DM2 typically has no interruptions within it. The mutant allele, the normal allele has interruptions, so the CCTG is interrupted by other sequences. But in the pathogenic state, those interruptions are lost, and the expansions can become quite great. And the actual range of involvement ranges from about 75 or 80 to over 10,000. And the mean is somewhere around 5,000. And very much like you see in classical DM1 myotonic dystrophy, you also see the same kinds of aggregates of mRNA in the nuclei. Oh, in this case, it's a CCUG repeat. What does this protein do? It's not a protein. It's not a protein kinase like the myotrophin is. This is expressed predominantly in muscle, but it's also expressed in brain. It's a single-stranded DNA binding protein. It's a regulatory element of sterol, and typically is cytoplasmic or in the endoplasmic reticulum. It has no relationship to the myotrophin protein, so functionally they don't do the same thing, and their surrounding genes are completely different. So despite the fact that these are totally unrelated genes, when they're mutated they cause a very similar clinical picture. And this is just sort of schematic of what the genes look like. This is the myotrophin, so this is the DM1 gene. So out in this 3' untranslated region, there is this expansion of the CTG that can range anywhere from 80 to over 2,000. And then in the DM2 gene, the zinc finger 9 protein, the expansion is in this first intron between exon 1 and 2. So what's the significance of these expansions? So there are some proteins that can bind to these CUG sites, and There is one called CELF, which stands for CUG-Binding protein and ETR3-like family, whatever the heck that means. And these are basically splicing factors. So these are proteins that are related to splicing of message. And the CELF protein binds to CUG repeats in the transcripts, but it doesn't seem to co-localize with the RNA inclusions. So we don't see that protein accumulating in those RNA inclusions. But there's a second protein called Muscleblind or Muscleblind-like. It's actually named after a Drosophila protein that does associate with these inclusions. And the CELF protein is typically seen during development and tends to be downregulated at birth, but not necessarily in the brain. And muscleblind, for the most part, most forms of muscleblind tend to be upregulated around the time of birth. So they become sort of the dominant type of splicing factor once you get in the post-fetal period. So if there is the normal transition from CELF to muscleblind, you get a different form of splicing in a number of different genes because the muscleblind now takes over. So in the absence of muscleblind, the CELF then can take over from the muscleblind, and then you have a persistence of the fetal pattern of splicing. So there's not a maturation of the protein forms that would normally be seen. And over time, we've discovered that there's a number of important genes that are affected by this, and one of them is a chloride channel, CLC1, and this apparently accounts for the myotonia. So if there's a mutation in this, or an abnormality of splicing in this gene, you get myotonia. Tau protein, which is the microtubule-associated protein that's very abundant in brain and very important in certain neurodegenerative diseases like Alzheimer's disease, also has alternative splicing. Troponin in cardiac muscle, myotubularin, insulin receptors, which may account for some of the insulin resistance in these patients. We know that if you knock out muscleblind, you get a phenotype that's very similar to myotonic dystrophy, both in muscle, and we're starting to see perhaps even in brain that there is some abnormality of brain splicing as well. And if you overexpress CELF in mice, you get a similar effect of muscleblind deficiency, so that the CELF overwhelms the effect of the muscleblind and becomes the dominant factor in splicing. So there's a persistence of this fetal pattern of splicing. This is kind of a schematic of how this might work. The CUG repeats, or the CCUG repeats for that matter, can form these hairpin-like configurations that appear to aggregate the muscleblind. So the muscleblind tends to associate with these inclusions in the nuclei of the cells. And it basically sort of sops up the muscleblind, leaving CELF to be the only interactor left. And so the alternative splicing is basically dependent on CELF rather. So it becomes alternative in the sense that you no longer have the adult pattern, but you're reverting to a fetal pattern of splicing.
Brent?
Yes?
Does that happen in wild type as well, but with less binding because the hairpin is smaller?
We don't see the inclusions in normal.
But do you get the...
Yeah, that I don't know. I mean, you're talking to the wrong guy.
In the normal situation, I thought that the normal size hairpins, when it's in a hairpin form, it binds one. When it's not in a hairpin form, it binds the other. And so when you form abnormal hairpins, it tends to bind muscleblind. Okay. And so then it sequesters them.
Okay. So it's basically depleting it.
Right. So it's competitive based on the three-dimensional, or I guess two-dimensional structure of the RNA.
Yeah, so I mean, the muscleblind is the one that tends to aggregate in these long hairpin repeats. And you can, it shows here both with the CUG and the CCUG repeats. You can see co-localization of the RNA and muscleblind. So here you see the RNA red, muscleblind green, and then the merged picture shows the yellow. So that's true in both myotonic dystrophies types 1 and type 2. So the alternative splicing can affect the proteins in different ways. With the insulin receptor, the adult form typically has an extra exon, which tends to give a higher signaling receptor, so a more functional receptor. If that exon is excluded, so if you have the CELF form of the splicing, that exon is missing, and you get a lower signaling receptor. So that apparently accounts for some of the insulin resistance when this form is present. The chloride channel is a little bit different in that in the adult form, actually there's exclusion of a particular exon, and then that makes the normal protein. And when this is formed later in life, it actually is considered to be sort of nonsense, and so is actually often destroyed. So there's essentially a loss of function of this chloride channel, and apparently that's what accounts for the myotonia that's present in this disease. Here's some of the proteins that have misregulated alternative splicing, and so cardiac troponin is one, insulin receptor, chloride channel, tau, which we talked about previously. Another brain protein is the NMDA receptor. So this is one of the common glutamate receptors in the brain. And glutamate is one of the major excitatory neurotransmitters in the brain. So this seems to have an alternative pattern. We don't really know how much effect it actually has on the function, but it's one potential target. Also of interest is the amyloid precursor protein, which is another protein that is abnormal in Alzheimer's disease. It tends to accumulate. But unlike in Alzheimer's disease, patients with DM1 or DM2 don't tend to have the accumulation of the A-beta fragments that are derived from this protein that are typically seen in Alzheimer's disease. And we'll get into that more when we talk about the pathology. And then in the last few years, there's a new player on the block. The importance of this, I think, still is somewhat open to question, but it's something that we're planning to look at in considerable detail. This work came out of Laura Ranum's lab several years ago in a paper that has more authors than the Minneapolis phone book, I think. But this is a concept known as RAN translation, or repeat-associated non-ATG translation. And the idea is that in these sort of hairpin-like loops, you can actually have initiation of translation that is randomly starts. So depending upon which nucleotide it starts at, you can be in 3 different frames. And actually, if you have it going in the antisense direction, you could have 3 additional different frames. So the potential is that you could have a polyglutamine, polyalanine, polyserine, polylysine, and polycysteine, polyleucine, I guess. And SCA8, which has a somewhat similar mechanism to DM1, she has shown that you can see accumulation of polyalanine using antibodies against polyalanine. And likewise in DM1 patients, she's been able to show polyglutamine accumulation. So what the role of these polypeptide sequences are in pathogenesis is still unclear, but it's another potential mechanism of injury to cells. Also in the DM2 patient, this wasn't in this paper, but it's some preliminary data that we had for a grant submission. They're also, because this is a tetranucleotide repeat, you actually get a sequence of 4 different amino acids, basically because it's not a triplet, it's a tetra repeat. So you get leucine, proline, alanine, cysteine, sort of a polypeptide composed of that tetramer of amino acids. And those are also present in DM2 patients, in certain cells at least. This is a very busy slide. This is just to point out this is some immunohistochemistry studies from SCA8 showing the polyalanine immunoreactivity in Purkinje cells, which is one of the target cells of this disease. And then in DM1, these are myoblasts are derived from patients with myotonic dystrophy, and you can see in the wild type there's no staining, and in the DM1 patients there is. This is actually in a transgenic model also, where you have an expansion of a CUG-repeat. So both of these, this is an extra long expansion, this is a relatively short expansion, but they both seem to accumulate the protein or the peptide. So we don't really know exactly what the role of these things are. But so to kind of summarize the molecular pathology, we know that there's mutations in two completely different genes. One is this protein kinase, and that's the most commonly mutated one that we see in myotonic dystrophy type 1. One is the zinc finger 9 protein, which is seen as the mutation in myotonic dystrophy type 2. So despite the fact that these genes are completely different in terms of their effect, or what their gene product is, mutations lead to similar pathological phenotypes. And they both have these repeat expansions in non-coding regions of the protein. They both make, they transcribe these sequences of CCUG or CUG, and that they are capable of sequestering this muscleblind-like protein. And that leads to the splicing abnormalities that appear to be the major cause of pathogenesis. However, both forms of the disease do have evidence of this RAN translation. So whether or not that plays a significant role in pathogenesis, I think, is still somewhat open to question. So let's talk a little bit about what's known about the neuropathology of myotonic dystrophy. And I could probably stop right here. There's a lot of talk, but not a lot of, there's a lot of smoke, but not a lot of fire, I guess. So what I'd like to do for a second is just talk a little bit about neuroimaging, because I'm going to be showing some neuroimaging, and I'd like to get you all on the same page. So these are MRI scans, and there's several different ways of looking at an MRI. And it's based on physics, which is I can't explain to you, but I can recognize it when I see it. So one of the sequences is called T1, and this sequence looks like a picture of the brain. The white matter is white, the gray matter is gray. The fluid is dark, so CSF is dark, so the ventricles look dark. The subarachnoid space, the sulci look dark because they're filled with water. So water looks dark, white matter looks white, gray matter looks gray. T2 weighted imaging tends to accentuate water, both in the CSF and in tissue. So it almost looks like a negative picture of the T1. So the white matter is dark because there's very little water in the white matter because of the myelin, which is very lipophilic. So hydrophobic, so water is not a significant component of white matter. The gray matter is still kind of gray, and the CSF is bright. Then they came up with a new technique back maybe about 20 years ago, primarily to look at patients with MS, which is called fluid attenuated inversion recovery, or FLAIR images. And this is basically a T2 image, but they have a trick which I don't quite understand how they do it, but they invert the water signal that's free water in the CSF. So instead of looking bright, it looks dark. But tissue water still looks light. So it's a very sensitive way to pick up pathology, particularly in the white matter, because the white matter, if it's damaged, tends to have a more hydrophilic nature to it because the myelin is probably damaged, and so there are places in the white matter now that are much more readily accessible to water, and so the water indicates that there's something going on. Sometimes it's very subtle, sometimes it's not so much, but the appearance radiographically can sometimes be much more impressive than the appearance pathologically. This is the sort of the bottom line. So there have been a number of studies using this technique, MRI, to look at patients with myotonic dystrophy. And these studies actually started 20 years ago or so. And what they did was they would take a number of patients with the disease and try to age match controls and just compare a number of different features. And one of the things they showed is that patients with myotonic dystrophy tended to have high incidence of white matter hyperintensity. So on their T2 weighted images, they had white spots in their white matter compared to controls who had far fewer in the same age distribution. There were certain areas that were kind of hot spots, one of which was in the sort of the anterior parts of the temporal lobes in the white matter just below the cortex. They also looked at gray matter, just kind of using morphometric techniques. Looked at the cortex of the using the scans, and basically they determined that there was cortical atrophy present in myotonic dystrophy patients, or at least more than there were in the control group. But what was interesting was that the cortical atrophy and the white matter hyperintensities didn't really correlate very well. And likewise, the presence of these abnormalities didn't correlate very well with the age of the patient, with the duration of the disease, with the amount of physical disability or the severity of neuropsych impairment. So it seemed to be kind of, not particularly firm connection between the changes that they saw by imaging and what the patients actually look like clinically. This is an example of some of the changes that are seen. Sometimes they're fairly subtle with just some hyperintensity around the ventricular system. Other cases, they're more disparate, fairly widespread, and kind of sort of punctate. In other areas, they can sort of line the ventricular system, not necessarily always symmetrical. And the nature of these is still somewhat in dispute. Sometimes they're actually true lesions. Sometimes there's really very little that you see microscopically. Unfortunately, many of these patients have never been studied, and many of the patients that have been studied pathologically, we don't have neuroimaging on. So we're kind of looking at an average of what is seen and trying to extrapolate that into what we see pathologically, and sometimes that can be difficult.
Excuse me,
Yes?
would the MRAs be abnormal in all of these patients, do you think?
The what?
MRAs, the vascular studies.
And they have not been. Actually, MRA is mostly a macrovascular technique, so it really doesn't pick up very distal circulation abnormalities. So I think if there are abnormalities in circulation, they are probably more distal, and MRA is not a particularly good technique for finding that. This just shows the pattern that I was talking about, the sort of anterior temporal lobe pattern. That can be seen both in congenital and in adult cases, although it's usually not seen in the DM2 patients. Another study looking at neuroimaging and myotonic dystrophy, they just compared normals and patients with DM1 and DM2, and they just looked basically at brain volume, how much of the intracranial space was occupied by brain and how much was occupied by fluid. And the normal index was about 85%. In the DM1 patients, it was fairly significantly decreased, and the DM2 patients were somewhat in between. There are some problems with volume measurements. I think that they don't always really indicate necessarily atrophy. Sometimes hydration state and things like that can make a difference. So I'm always a little concerned about equating volume with atrophy. This is basically several other studies that have been sort of a synopsis of several other studies. In one, both brain and cortical volumes were significantly decreased in the DM1 patients, but the white matter volumes in this case didn't seem to be any different, even though there was a trend toward that possibility. Another fairly common hotspot is in sort of the posterior parts of the ventricular system, around the confluence of the ventricular horns, and that can be seen both in congenital and in the adult onset. And then another finding that's been seen pathologically is what appears to be accentuation of perivascular spaces in the white matter. So some of these white matter punctate changes appear to be dilatations of perivascular spaces, which again would give a water-like signal. Another technique for looking at the brain is a more recently developed MRI technique called diffusion tensor imaging. And this is rather complicated, and I'm not going to spend a whole lot of time trying to explain it. But basically, it's a way that you can look at the integrity of fiber pathways within the brain. And it's very nice for sort of mapping fiber tracts. So you can kind of look at things in the X plane, the Y plane, and the Z plane, and kind of follow the tractography of the white matter in particular. And it depends largely on the fact that the white matter is very organized. So the fibers, so water tends, in the normal state, would diffuse in all directions, but it has some restriction of its diffusion by the anatomy. So it'll tend to diffuse along the long axis of how the fibers run. And if that long axis is somewhat disturbed, then you will have what's called a decrease in anisotropy. So anisotropy means that it's tending to conform to the anatomy. And so if the anatomy is somewhat disrupted, that will be lost. So what's been seen in a number of studies of patients with myotonic dystrophy is that if you do this DTI imaging, you see that there are abnormalities in the white matter that don't necessarily correlate with the T2 hyperintensities that you see using classical MRI. So there's something different that's not necessarily related to these things that we identified focally using classical MRI. And one of the places that seems to be a bit of a hot spot is in the corpus callosum, particularly the posterior corpus callosum. And there was a study done in Japan a few years back where they claimed that the areas where the corpus callosum was most heavily affected were in areas where there seemed to be lower volumes in the cortex. So the projections from those cortical areas would be accounted for by those areas of corpus callosum. One of the problems with diffusion tensor imaging is we don't really understand the nature, and I'll get back into this, we don't really understand what causes these abnormalities. You'd think it would be just like demyelination or axon loss and things like that, but sometimes it occurs where you don't really see that very well, and sometimes where you have demyelination, the changes in the tensor imaging are considerably less than you might expect. So I think it was once thought that there's a pretty tight correlation between these changes and true neuropathology, and I'm beginning to have some doubts, or at least if it is, it's much more subtle than what it has been thought. So there's been very few studies that have actually looked at patients who have had imaging studies, and this is one study that was actually done a number of years ago by a Japanese group, and this was a patient who had changes, this classical changes in sort of the subcortical areas of the anterior temporal lobes. When the patient died, they examined these areas and they saw pallor of the myelin. This is a myelin stain. It's in black and white because that's how it was published. But you can see that there's pallor here. So there's a diminution in the myelin density. Whether it's actually due to demyelination, I think, is not entirely clear, or whether it's just sort of an attenuation of the overall white matter. This is a higher power figure, just showing that there's a lot more space between the fibers. Normally they should be very densely packed. And if we did a myelin stain of normal white matter at this power, it would be fairly dark looking. It would be a dark gray picture. So there's a lot of space, which would give you a water-like signal. These spaces are occupied by water. So when you do the T2 weighted imaging, you should see that. So that is essentially the correlation that's seen.
No inflammation.
What the significance of it is not clear. No, there's no inflammation. It doesn't really look like, they didn't really study it as far as I can recall with axon stains versus myelin stains, but it doesn't really look like a classical demyelinating lesion. I'm just going to kind of walk through a list of things that people have seen in various published reports of the neuropathology of myotonic dystrophy. And it's not terribly impressive, frankly. So the white matter rarefaction, like I just showed you, is one thing that was shown in 1 or 2 reports. And I've seen it in a few of the cases that we've studied as well, but it's not a consistent finding. The dilated perivascular spaces, I've seen it in a few of our cases. Again, not a consistent finding. And I rather doubt if it has any real clinical significance. One paper showed neuronal loss in the superficial layer of the frontal and parietal cortex with extensive neuronal loss in the occipital cortex. I have never seen that in any of our cases. Occasionally you'll see some cortical gliosis, but most of the time it's very hard to tell a patient with myotonic dystrophy from a patient who has nothing at all. And to me, that's kind of a guideline, is that if you can take a tray of slides from one patient, a tray of slides from another patient, and pick out the disease, then you've got something. If you can't, then you don't have something. Another thing that's been found, and this is something I'm going to talk about in a minute or two, is that they have increased numbers of neurofibrillary tangles. And they seem to be sort of pre-senile. They come on earlier in life than you typically see with aging, but they're often in the same distribution that you see with aging. but they come on 10 to 20 years earlier. And so they're primarily seen without senile plaques. So it doesn't really look like Alzheimer's disease. It's the neurofibrillary pathology without the plaque formation. And then there was a lot of noise made about these inclusion bodies in several areas of the brain, including particularly the thalamus and substantia nigra. And I'll show you what those look like in a second. And I'm not very impressed with those either, because you can see them in normal people if you look hard enough. Another paper, this is a great example of salami publication. They published the same 8 patients in 3 different papers. So they took each nucleus and published it as a separate paper. So a little CV synthetase there. But anyway, this was a group in Japan. And the point that they made was that there were 3 patients with myotonic dystrophy out of 8 who had hypoventilation. So that's another problem that they can get. In addition to hypersomnia, they can sometimes have problems with hypoventilation. That's thought to be a central hypoventilation. And there were 3 nuclei that they looked at, and they claimed that they saw decreased numbers of neurons. There's I think some methodological problems with this, and I'm not sure they did it blinded, which always bothers me. When you're looking for things that will explain things, you can often see things. And it's hard to do these kinds of studies in human material to actually do accurate counting of nuclei that are actually quite ill-defined. So I have some reservations about that as well. There have been some reports of just cortical laminar abnormalities, so like the cortex isn't arranged correctly. This was mostly very early papers written back in the 60s, and that's something that I have not seen in any of the cases that I've looked at either. It is clear, particularly in the congenital cases, that there's often ventricular dilatation, so there does seem to be some degree of brain atrophy. And I think in the congenital cases, it's usually fairly prominent. And then splicing abnormalities in tau have been shown. So actually people have developed antibodies against certain exons that are left out of the the fetal form of splicing. So in patients with DM1, if they use those antibodies to try to label the neurofibrillary tangles, they don't stain anything. So the tangles are composed of this abnormal tau. So this is an example of the perivascular space dilatation. This is at low power, at higher power. The tissue around it looks entirely normal. So I doubt if this has really any pathophysiological significance. And as I said, it's found in occasional patients, but certainly not a widespread finding. These are the dreaded cytoplasmic inclusions. They're these little eosinophilic bodies. The cells that contain them look perfectly happy. In the areas in which they occur do not have cell loss. They don't have any symptoms referable to dysfunction of those areas of the brain. So I think they're probably not of any significance whatsoever. And if you look for these in normal brains, you see them. Probably not to the same extent, but I rather doubt that they have any major significance. There was a paper came out a number of years ago by the group that originally described these, showing that they tended to stain with ubiquitin and to antibodies against microtubule-associated proteins 1 and 2, but negative for lots of other proteins, including the kitchen sink. One thing I alluded to earlier was the presence of the muscleblind containing inclusions. That certainly has been seen in muscle, but it's also been seen in brain as well. So Maury Swanson's group, in conjunction with a group in Rochester, did some studies looking at brain, and they showed the same thing where you have the RNA inclusions in the nuclei that co-localize both with Muscleblind-like 2 and Muscleblind-like 1. So these are both muscleblind proteins that are heavily expressed in brain really throughout life. And I pretty much have kind of summarized that already just when I talked about. Just to sort of reiterate the, actually one point I want to make is that the muscleblind immunostaining, in the absence of the inclusion, it tends to be fairly diffusely positive throughout the nucleus. but in the cells that have inclusions, it tends to be concentrated in the inclusions, like it's basically sopping up the free muscleblind. So you don't see the diffuse distribution in the cells that have the inclusions. But just to kind of reiterate some of the brain-specific proteins that are affected by the alternative splicing, including tau, the NMDA receptor, which may be important in learning function. It's very heavily expressed in the hippocampus. This may be more important in congenital myotonic dystrophy, perhaps, than it is in the adult form. And then the beta amyloid precursor protein, which we know is involved in Alzheimer's disease, but doesn't really seem to accumulate in patients with DM1 or DM2. So we decided to just take a look at tau pathology based on the material that we had, and primarily we looked at the immunohistochemical presence of phosphorylated tau in the hippocampus and in the entorhinal cortex. And so basically, just to get you oriented, the hippocampus is this structure here. It looks like a seahorse. And the primary pathology usually is in this region called the CA1 region. And then there's another area out here called the entorhinal cortex, which projects into the hippocampus. And this is another area that's commonly affected in both myotonic dystrophy, but also in Alzheimer's disease. So these two areas tend to be affected in Alzheimer's and in myotonic dystrophy. And as we age, we tend to develop tangles there. Whether that's a precursor of developing Alzheimer's disease or whether that's normal aging is still somewhat up in the air. But my guess is it's really sort of the early stages of Alzheimer's disease. But in Alzheimer's disease, you also develop senile plaques in these same locations and in other sites. And in patients with myotonic dystrophy, they have no greater accumulation than the normal population does. So we took some of our patients. This was one of our younger patients. Patient was 40 at the time of death. And this is from the entorhinal cortex. And you can see that there are occasional neurons that are staining. Some of them have kind of a tangle-like appearance. And there are some neuropil threads in the background. So these are axonal processes, presumably, that also contain accumulations of phosphorylated tau. As the patients got older, we started to see more. So this is a 56-year-old patient in that same region of the brain. And we can see some that have the appearance of actual tangles. So these would be silver positive using silver stains. But some of the cells just had immunoreactivity, probably in a pre-tangle accumulation of tau. But you also can see an abundant number of these neuropil thread-like structures in the background. This is a 66-year-old patient, again, with most of the neurons now expressing the tau protein, but not necessarily always in the form of tangles. And neuropil threads are somewhat apparent here.
What would you see in a normal age? Oh, never mind.
Here you go. You want to go to work for me? Yeah. Okay. So this is a patient that we took some slides or some sections from the NUN study, and this is an 80-year-old patient who was cognitively normal at the time of death. Had neuropsych testing within 1 year prior to death. And you can see occasional cells that have some immunoreactivity and a little bit of neuropil thread activity, but really not. So this is kind of a synopsis of it, just showing the results both with DM1 and DM2. So there tended to be quite a few neurons in the entorhinal cortex that were positive, particularly as the patients got over the age of 50. But even in their 40s, you were starting to see them. And with the DM2 patients, we saw a little more, but these patients tended to be older. They tend to live longer because they don't have as severe a disease. On the controls, the young controls, you really see essentially nothing up until 60s or 70s. And then even in the people in their 80s, these were all from the NUN study, and they still had relatively mild involvement. So there's kind of a precocious accumulation of tangles that it seemed to be, kind of in the same distribution as you see with aging. And I pretty much have summarized that, I guess. So what does this mean? Probably not much. Because, people that have, we have an 80-year-old nun who has some changes in her entorhinal cortex, and she's cognitively intact. And the kind of changes these patients have really don't relate to memory function. So this area of the brain is really largely memory related. So the problems that AD patients get early in the course of disease are usually memory type problems. So I don't think that this is very significant in the overall picture. It's something we can do, something we can look at, but what does it really mean? So another thing we decided to do is to look at the white matter, particularly the corpus callosum, based on these diffusion tensor imaging studies. And so we went back and found that area of the brain, actually resubmitted from the cases we still had wet tissue. The reason for this was because of these hot spots that were shown on the DTI studies. So we looked at the posterior corpus callosum, and we used H&E, we used Luxol-Fast Blue, which is a myelin stain. We used immunohistochemistry for CD68, which is a macrophage microglial marker, and GFAP, which is a marker for astrocytosis. And so I had these evaluated blindly, and we didn't see any difference. We couldn't pick the patients out. There were occasional patients who had some white matter changes, but they were equally present in the controls and equally present in the DM patients. So whatever these diffuse and tensor imaging changes are, we don't really, I don't know how to explain them. The one caveat is that none of the patients that we studied had DTR, so we don't know if they had those lesions. But if you look at a population of patients, the vast majority have changes in those areas. So my guess is that if they were studied, some of them would have had changes in those areas. So that comes down to the question to what do these changes really represent? I mean, it's always been assumed that they're structural alterations. So is it axonal loss? So I mean, the idea that Japanese group had was that, well, there's cortical atrophy in areas that are projecting into areas that are affected. So that means that they're losing the cell bodies and maybe losing the axon. But typically when that happens, you see some effect in the white matter. You see gliosis, you may see macrophage. You see some indication that there's been fiber loss, and we're not seeing that. Myelin loss, similarly, if there's myelin loss, you usually can see it. Now, could it just be attenuation of myelin in some way? That would be very difficult to evaluate, perhaps, I don't know. Or could it be attenuation of axonal diameter? And this is perhaps a more likely possibility, that perhaps cell bodies are becoming somewhat atrophic, reduced in size, and that could reduce the volume of the cortex without necessarily having the cells drop out. And perhaps then their axons would be reduced in volume or size, and that could have some effect on the anisotropy of the fiber tracts that are represented by those fibers. Could it be something unrelated to the actual integrity of the fibers themselves? Could it just be increased tissue water due to vascular changes? I mean, so things like that could potentially have an effect as well. We have no evidence that there's anything wrong with the blood vessels in these patients, and we don't know of any abnormalities in splicing that would have an impact on the vasculature, at least at this stage of our knowledge. So to summarize the neuropathology at this point is that we don't know very much. So it's really very poorly understood. We think that the congenital form is largely maldevelopmental, and there may be some additional neurodegenerative changes that occur throughout the lives of these patients. They do tend to get greater ventricular dilatation throughout life. And that the adult form is probably neurodegenerative or perhaps just functional. Maybe just the fact that you have, very similar to the myotonia, you have abnormal receptor function that is primarily a pathophysiological disease and not so much a structural pathology disease. So one of our goals is to try to identify additional proteins with splicing abnormalities in these diseases that might give us some clue to what's going on and may help explain some of these sort of mild structural hints of structural changes, at least from the imaging, and perhaps get a better handle on what the functional changes are. So we will continue to do both the conventional MRI studies and the diffusion tensor imaging studies on our patient population, and at some point we may get some patients who've actually had the studies come to autopsy so we can do a more direct correlation. So I just want to spend a minute or two talking about sort of ongoing and future studies. I'm very happy to announce that we just got a renewal on this program project after we submitted about a year ago and didn't get funded, and we were submitted very quickly and we got funded as of the 30th, I believe. So maybe we'll even get paid, I don't know. But this is a program project that's probably the most widespread program project in the history of the NIH. We have PIs in Gainesville, Florida, in Palo Alto, and the core is at the University of Minnesota, which is kind of weird. It started off with two of the PIs being here, but they've both moved on. So we have teleconferences that span 3 time zones, so it's often very hard to get people together. But so Tim Ebner and I are the sort of the local presence. And I just want to talk briefly about that. What's going to be done over the next 5 years, or attempted to be done. So Laura's going to focus a lot on the RAND translation, because that's, I mean, she found this stuff and now she's, you know, it's like she has a new toy. So she's going to spend a lot of time looking at the RAND translation, and to see how much, you know, how much of this stuff actually accumulates in the brains of patients and in some animal models, and then see if it really plays a role in pathogenesis. Maury Swanson, who is at Florida, he's been very interested in the RNA, you know, aspects of it. Very interested in finding new targets, trying to identify splicing abnormalities and which proteins may be abnormally spliced, which can be very helpful in terms of trying to determine where we would focus, where we would look in the brain, what kinds of things might be important in disease pathogenesis. So he's doing this both on human tissue and on some knockout models that he has. We've tried to make models of the CUG or CUG repeats, but we've had very difficult time getting good expression in the brain, so we've kind of abandoned that approach in our recent studies. And Maury's actually had some pretty good luck using the muscle-blind knockout approach, which mimics to a great extent what you see in the human diseases. So, so he's going to be concentrating on that, both using the muscle-blind knockouts and human tissue that we get from autopsies to try to identify these splicing abnormalities. He's also developing muscle-blind 2, muscle-blind 3 knockouts. So muscle-blind 3 is a form of the protein that's expressed almost exclusively during embryonic development and tends to pretty much shut off by the time the animals are born. So he's hoping that perhaps this could lead to a model of more of the congenital form of the disease, but this is still kind of in the early stages. And then John is out at Stanford now, and so they have a very fancy sleep lab out there, so he's been collaborating with people in their sleep studies. So he's going to be concentrating to a large extent on some of the sleep abnormalities, trying to distinguish differences between the patients who have sleep abnormalities and those who don't. But in addition to that, he's going to be continuing to look at the white matter imaging, both using conventional MRI and the diffusion tensor imaging, and trying to correlate that better with the neuropsych status. So they'll be doing batteries of neuropsych testing and evaluation of the neuroimaging. So the role that Tim and I play is, Tim's primarily involved in looking at the animal models, and he's doing neurophysiological studies to try to complement some of the findings that are seen behaviorally. I'm involved in the neuropath imaging, or neuropath examination of animal models, as well as continuing to look at all the human patients that come through. And hopefully, once we get a better handle on some of the splicing targets, maybe we can focus our examination more on certain protein localizations, things like that. Or if we can determine, you know, which proteins are abnormally spliced, we can look for their distribution in the brain and see if those brain regions tend to be affected more than, so if you can focus your exam somewhere, you may be able to find subtle differences that with a more generalized exam you would just miss. And then we'll also be helping Laura with the RAND translation project in terms of localization of these abnormal polypeptide products that are generated by the RAND translation. So just to acknowledge people who've been involved in these studies. So Laura Ranum, who was formerly here, actually was a postdoc in Harry's lab originally, started her own lab, and been very productive both in the ataxia field and myotonic dystrophy. Maury Swanson, who's really been one of the key players in the myotonic dystrophy field for the last couple of decades. John Day, who's a neurologist who was formerly here. And of course, Tim Ebner in the neuroscience department, who's been a long-time collaborator. In my lab, Lou Ann Anderson, who's my principal technician. There was a woman, Miriam Karad Pesho, who was a volunteer in my lab several years ago, and she did help me with some of the studies, particularly the white matter studies. Joyce Mintz, who's another histotechnologist who helped us, and then Kenji Takamura, who was in John Day's lab, has done a lot with us, particularly in the autopsy procurement. And that's it, if there's any questions. If anybody has a roll of quarters, we can send it to Congress. Any questions? Great. Thanks.